So thank you. It's a pleasure being here. I appreciate the invitation. I have, as you heard, I'm a recent arrival within the last year. I learned this morning that my honeymoon period is over since my dean and chancellor, as words of encouragement, told me not to screw up. So I'm assuming that means that I'm now fully here. So I'm a pediatric oncologist and a translational scientist, and I've been involved in sort of the development at the preclinical level of immunotherapy for cancer, sort of as a concept. And I slightly changed the title of my talk to sort of CAR T-cell therapy, which is the therapy I'm going to talk about, and what have we learned so far. And for those of you who would like to sleep and not pay any more attention, I'll tell you exactly what we've learned. We've learned a lot, and we've learned that there's a lot that we don't know. And so hopefully I'll convince you of that today. The other thing that I certainly have learned as a part of this experience, it's really been a privilege in a way. I mean, obviously, these sorts of therapies, build upon decades of scientific progress, and there's lots of folks that deserve a lot more credit than I do for this. But it's really been a privilege to be a part of a therapy, really in its infancy, and to kind of watch that evolution. And I'll tell you a bit about that journey. So just by way of a quick outline, I think it's always important to kind of go back and look at history. It tells us a lot, and it's always humbling to kind of see where we come from. And then I'll talk a bit, just by way of introduction, about what a chimeric antigen receptor T-cell is. And I'll sort of then give you the somewhat remarkable initial clinical experience with CAR T-cell therapy. And then at the end, I'll get back to, I think, the challenges and hopefully at the end have some questions about, what we might do about that. So just as a reminder, I mean, the concept of immunotherapy is old, right? So it really began at the turn of the last century. A physician by the name of William Cooley, who was at a hospital in New York City that ultimately became Sloan Kettering. And he was a surgeon, and he operated on sarcomas. And in fact, one of his claims to fame was that he operated on a friend of J.D. Rockefeller's, a teenager who had an aggressive sarcoma. And he made this observation. It was a time when certainly antisepsis was known, but there weren't a lot of antibiotics around, none. And the patients developed postoperative infections. And he had patients where he knew that he didn't completely resect the tumor, and yet in the setting of a postoperative infection, they had spontaneous regression and did not recur. And this was what I show up there is a first page of one of his papers. Papers were much more complicated then. There were multiple parts. But the last part was the treatment of sarcoma by inoculation with erysipelas. And essentially what he was doing was injecting infection, pus, into tumors. And he had responses with this approach. The problem is that they were very unpredictable because they didn't understand the science. And at the same time, there was another physician at the same hospital. His name was James Ewing. He gave name to the Ewing sarcoma, a pediatric sarcoma. And he was developing radiation treatment for cancer, and the responses to radiation were much more predictable. And there was a bit of a battle between the two of them in terms of which approach made the most sense for sarcoma, and the predictability of the responses to radiation, ultimately won out, and that was the one that went forward. And really at that point, immunotherapy sort of died in a way as a field. And this is one of his quotes, which is, "Nature often gives us hints to her profoundest secrets." And this is just a picture of a patient that he treated who actually was cured with this treatment. So, I mean, it really was probably 50 to 60 years from sort of that event at what was eventually to become Sloan Kettering to, that battle at Sloan Kettering to the 70s, really, with the advent of bone marrow transplant, IL-2, that we really saw a resurgence of immunotherapy for cancer. And it really was due to the fact that the field began to understand a bit about what was happening. So just by way of primer, and there was a nice introduction this morning to sort of the complexity of T cell receptor and B cell receptor recognition of antigens. But just remind everybody that the T cell receptor, in addition to having this very exquisite specificity based on rearrangements of genes, that in and of itself, that activation is in and of itself is not sufficient to really make a T cell do what it is endowed to do, which is to kill target cells. It requires this complex network of other signals that really drive the full activation of the T cell. And the other thing that was learned sort of through this process of looking at co-receptor signaling in T cells was that there was this whole other set of receptors that are essentially brakes on the immune system. And so these are intended, so when one develops influenza, most of the symptoms associated with influenza are not due to the flu itself, but it's the immune response to the flu. And so it can be a fairly violent response, and there's this set of brakes that are essentially intended to cool that off at the end. And these are the checkpoints. And so I think most of you are probably familiar with really the success with immune checkpoint inhibitor treatment for cancer that was really pioneered by Jim Allison, who along with Tasuku Honjo won the Nobel Prize for these observations. And I actually was at the NIH in the early 90s when I saw Jim Allison present this data. And this is demonstrating that if you block the brake on the immune system, that you can get eradication of tumors in mice, right? And at that time, it was a science paper, it was an important observation. But quite frankly, a lot of us were doing pretty well treating tumors in mice. We weren't doing so well using the immune system to treat tumors in humans. So as with other therapies, though, this ended up going into the clinic, and it turned out that it was actually quite effective in some patients. And so this is a paper from the surgery branch at the NCI showing eradication of tumors in the lung, those white dots that you see there, and in the brain in patients who received these checkpoint inhibitors. And those of you who remember Jimmy Carter had a liver tumor and then had a brain metastasis. He received one of these checkpoint inhibitors and had a complete remission. So really dramatic responses in some patients. And this then resulted in really an explosion of immune checkpoint inhibitors for the treatment of cancer. And this is a review from Science, Tony Ribas and Jed Walchuk. And so what you see up there in blue is the first patients being dosed, and then in red is the FDA approval, and you see all the indications over there. So certainly really, so this timeline goes from 2006 to 2018. So it's really been a remarkable explosion of agents, these so-called immune checkpoints. And I think many would say has really transformed the treatment of some types of cancer. So unfortunately, and as I mentioned, and I think we saw data earlier today, that not all patients respond. And the response rates vary depending on the subtype of cancer that's being treated. But maybe about 20, 30% or so of patients respond. It is true, though, that the patients who do respond, it can be dramatic, and they can actually be cured. There are patients that are long-term survivors after receiving immune checkpoint inhibitors. But there are other patients where the immune checkpoint inhibitors have no response. And one of the observations we heard this morning about the repertoire diversity of the T cell pool in a patient being a predictor of response, that is sort of the spectrum of T cells that can see different antigens, as you would imagine, might predict a patient who would respond to this therapy, because this therapy really relies on the ability for the endogenous immune system to, quote, see the tumor, right? There has to be recognition of the tumor that's pre-existent for these therapies to work. But one of the things that we're learning is that the other issue is on the tumor side. So we know that that tumors acquire multiple mutations generally as part of their development, but the number of mutations sort of can range quite a bit. And this is a progression-free survival curve with immune checkpoint inhibitors, and this is broken down based on percentiles of the mutational burden of the tumors. So the patients who have a higher mutational burden, shown in blue, are more likely to achieve a response and to benefit from the therapy. Patients that have mutationally bland or more bland tumors tend not to respond. And we know that those mutations are in some ways what allows the immune system to see the tumor. Unfortunately, the number of mutations per megabase, or the number of mutations, the amount of mutations in a tumor, it varies quite a bit, and it's not an accident. So this is just a slide. Each of those dots is an individual genome, different subsets of cancer, and obviously the higher they are, the more mutations. So on the far right in blue, the arrows I highlighted there, those are the tumors that tend to respond to immune checkpoint inhibitors. So I'm a pediatric oncologist. All the tumors that I treat are on the far left. And it is absolutely the case that the responses to immune checkpoint inhibitors in pediatric tumors has really been dismal. I mean, there's been almost no activity in most patients. And so this therapy, although it's really been transformative for adult epithelial cancers, has not really done much for the field of pediatric oncology. So what's the other way that we can go about harnessing the power of the immune system? So as I said, and as we heard earlier today, T cells, rearrange these receptors that gives them exquisite specificity for antigens. But the other possibility would be for us to control what the T cells are able to see by genetically modifying them with another receptor that gives them recognition of an antigen that we desire. And so that is the field that I'm going to talk about today, which is essentially removing T cells from patients and endowing them with a different specificity. And although there's been success with the endogenous T cell receptor for sure, in pediatric cancer, much of the success has been with these chimeric antigen receptors. And the structure of the CAR is shown on the right there, and they're called chimeric because the binding specificity is generally endowed by an antibody, and the nice thing about antibodies is they see surface targets and they see them free of MHC restriction, which is one of the potential limitations of TCR-based immunotherapy, and then linking that antibody specificity to T cell receptor signaling machinery, which is shown in blue and green in the slide up above. So chimeric receptor antibody, B cell based, signaling T cell based. And so these are genetic constructs that can be introduced into T cells ex vivo and then infused back into patients. So just another piece of history. So obviously we've seen dramatic success, but this sort of a field obviously takes time. And I like to go back to the original, really two publications that describe anything that resembles really a chimeric antigen receptor. And so the one on the left actually was a paper that came out in 1987, and it described constructs that resemble the CAR. And it was an amazing paper. It was literally typed, it was in a throwaway journal, typed on a typewriter, it looked that way, and all of the figures were hand-drawn. It was really remarkable. And then on the right was 2 years later, Zelig Eshar, who often gets the credit for having developed this technology, published his first paper with the construct shown down below. So in 1989, that paper, and then fast forward to 2010, so this is the first report of a dramatic response in a patient with refractory lymphoma at the surgery branch at NCI, Jim Kokendorfer's paper. So this was an individual who had chemotherapy refractory B-cell lymphoma, received CD19 targeted CAR T cells, and had a complete remission. And as I understand it, this patient is actually still in remission after receiving CAR T cells. So really a dramatic response. So now I'm going to just sort of turn my attention to the sort of initial experience beyond that first case report with CD19 targeted CAR T cells. So CD19 is a protein that is expressed on the surface of normal B lymphocytes as well as B cell malignancies. And in some ways, it was an ideal first target because in addition to eradicating tumor cells, this therapy also eradicates normal B cells and patients become B cell deficient. We can support patients by giving them immunoglobulin, so in some ways it's a somewhat dispensable target, but as I'll tell you in a little bit, that probably isn't going to be the case when you get into other tumors. So the way this therapy is manufactured is that patients undergo what's called an apheresis, which means we collect lymphocytes, T cells from the patient. This is right now a patient-specific product, which is a pretty remarkable thing considering it's FDA approved. The patients undergo an apheresis. These cells then go into culture and they get expanded in a dish, basically, in a manufacturing facility. And then we typically use viral vectors to introduce the chimeric antigen receptor gene into these T cells. If you Google using HIV to treat cancer, this is what will come up because the viruses that we use to introduce the gene are actually HIV-based viruses, lentiviruses. These cells then undergo subsequent expansion and then they're reinfused into the patients. So on the right is data from the NCI, from a trial that we conducted at the NCI with a CD19 targeted CAR T cell in pediatric leukemia that also expresses CD19. And we're happy in a phase 1 study if we see a 10 to 20% response rate. So that's what's called a waterfall plot. Each of those bars represents an individual patient. And what it's showing is change in the amount of leukemic cells in the bone marrow from baseline at 1 month following infusion of CD19 CAR T cells. So everything that hits minus 100% is a complete remission. So across multiple trials, the remission induction rate with this therapy was about 70 to 90% in the phase 1 experience, really a pretty remarkable experience. The other thing we learned was that all those hashed lines in the bar graphs represents what's called cytokine release syndrome. So essentially, as you would imagine, if a patient has a successful response, it's typically associated with inflammation, and that's the CRS, which can be quite toxic and is another challenge in the field. And certainly sometimes that can be severe enough that patients will actually not survive the therapy. So it's certainly something that we're having to deal with. The other thing that was pretty remarkable was that there were probably 6 different phase 1 clinical trials around the United States with CD19 CAR T cells, which is amazing. And they were all slightly different. The constructs were slightly different. The binding domain, there were 2 binding domains out there, and there were a couple of different signaling constructs. Just look at the red and the blue. The details don't matter too much, but they differed really in the co-stimulatory domain that was in the CAR construct. And the remarkable thing was they all worked. And I think, as I'll get to in a bit, I think we were a bit fooled by that because it turns out that CD19 is a fairly forgiving antigen. And as we've gotten into other antigens and other tumors, the details of the CAR construct are starting to matter more. And I'll come back to that in a little bit. So this was the data from the licensing trial for CD19 CAR T cells conducted by CHOP and Novartis. And this is pediatric ALL. These are patients who are all chemotherapy refractory. And there was a 90% remission induction rate. So pretty remarkable. It held up in the phase 2 study. And certainly very successful. But when you look at that survival curve, and what that shows is probability of continued remission in patients who achieve remission. And it is a perfect example of glasses half full, glasses half empty, right? So we know these patients are in remission at a month, but this really looks differently at what happens when you follow them further out. And what we're learning is that probably 50% or more of those patients will ultimately relapse within the first year after achieving remission. And what we're learning is that the patterns of recurrence really fall into 2 different categories. So one category is early, and it's because the CAR T cells are infused. They clear the tumor, and then the CAR T cells disappear, and the tumor comes back. So this is really a living therapy, and what we're learning is that persistence of these cells in the patients as a way of maintaining remission seems to be important. The other pattern of resistance that is really a bigger challenge in some ways is the later recurrence, and that is recurrence associated with loss of the CD19 protein on the surface of the leukemia cell. So the tumor essentially modifies itself, so it's no longer seen by the CAR. And that's a dot plot, flow cytometry plot. Each of those dots is a bone marrow of an individual patient that receives CD19 CAR T cells at the NCI. And as you can see, there's very little expression of CD19 after infusion of CD19 CAR T cells. So certainly, success, but certainly we're learning that there's a bit of, there are some holes in it. So, we were, with the initial experience of CD19, CAR T cells, we at the NCI started to think about other targets. We didn't really know about the loss of CD19 as a problem at the time, but we had a program focused on another protein that was expressed on B cells and B cell malignancies, and that's called CD22. And so we developed a chimeric antigen receptor that targeted CD22 and opened a clinical trial in 2015. Just as an interesting aside, so when I was the sponsor of the original IND, and as part of the approval process for that original IND, there's multiple layers of approval that are required. There's institutional, there's FDA, but this is a biologic therapy, and there's another group called the Office of Biologics Activity, and they have a committee called the RAC, and I will tell you that going to that committee is very much like being on the RAC, I would imagine, but it meant that I had to basically go and present my case as to why I wanted to open this trial. And an interesting discussion. So the first FDA approval for CD19 CAR T cells was pediatric ALL. That's an unheard of event, right? I mean, in pediatrics, we almost always get therapies handed down to us from our adult oncology colleagues. But the first indication was pediatrics, which is, again, remarkable. So when I took this to the RAC, I was told that I couldn't conduct the trial because I intended to include children on the initial cohort of patients that were treated with the CD22 CAR T cell. And the general paradigm is that you have to complete an adult trial before you open a pediatric trial and treat your first pediatric patient. That is fortunately changing. And the rationale is that children are vulnerable subjects, and since they can't consent for their own treatment, they're not able to really participate in those discussions. And parents are viewed as not being able to consent for them really in this sort of therapy. They have to sign the consent, but it's not felt that they can do this, unbiased, if you will, because they have that they're vulnerable because they have an interest in trying to do something for their children. So that's been the model in development of therapies for pediatrics. Fortunately, and I stood there for an hour and got raked over the coals for that. And at the end of that meeting, it's open for public comment, and a number of the pediatric cancer advocates, parents, knew that I was presenting this at the RAC, and they all showed up, and they took the microphone afterwards, and I just sat down and for an hour watched how they were sort of incensed by the implications that they couldn't consent for treatment for their children. So ultimately, we ended up getting this through. We had to treat adults first, but this experience that we've had, now we're up to our 65th patient on this trial right now, and almost entirely children and young adults in the trial. We've seen, as with the CD19 CAR, a pretty remarkable response rate, or about 75% remission. And importantly, we're almost entirely, exclusively treating now CD19 negative patients. So these are patients that receive CD19 CAR T cells, go into remission, relapse with loss of CD19. We treat them with a CD22 CAR T cell, and they go back in remission. So it's really been pretty remarkable. But as with the CD19 experience, the story certainly doesn't end here. So this is a curve on the left showing individual subjects and sort of how long they stay in remission. And what you see is that, so a lot of those patients aren't further out because that's how far out from therapy they are. But just pay attention to those black dots at the end. Those are patients who relapse now with leukemia that, in addition to not having CD19, now drops CD22. So what we're learning is that the patterns of resistance are getting pretty complex, and that's true both for leukemia and true for lymphoma. So again, I think the CD22 is very effective at putting patients back in remission, but again, we're not seeing durability because of cancer resistance. So the other thing, just a couple of editorial comments about running trials like this. It's really been remarkable because you learn from these experiences that some of the things that you wouldn't have predicted up front, sort of emerge as problems. And so I mentioned that all of these processes to manufacture these cells in the CAR constructs are different, but yet they all work. But in this trial, we slightly modified the manufacturing platform after the first 30 patients. Very minor manufacturing change. We thought it was going to make no difference. The FDA agreed. We went back into patients. We treated patients. Started seeing more toxicity. Had to go back, redo the safety phase, dose de-escalated, and then continued at a lower dose. A very minor change to manufacturing. Would have never predicted that would have had a difference. The other thing we've learned is that since this is a genetic modification and it goes into the host genome, it integrates in the host genome, that where it lands in the host genome can have an impact on how well these cells expand in patients. And so, again, just a plug for correlative studies in the context of these clinical trials, because we learn a tremendous amount by going back to the patients and these samples from these patients to really learn about what we're doing in this early experience. So I'm just going to now, at the last 5 minutes or so, I just want to talk a little bit about kind of where I see the future of this going. And I'll remind everybody of the two patterns of resistance that I mentioned. One being this what we call CAR T cell failure, where the CAR T cell maybe eradicates the tumor, but then it disappears, and then you have recurrence of leukemia that still has a target antigen. But then this other more complex pattern of resistance where the leukemia becomes resistant and drops the targeted protein on the surface. And so we began to think about, now that we knew that we had two good chimeric antigen receptors targeting two different proteins, CD19 and CD22, we know that by giving them sequentially, we can put patients back in remission, but they don't stay in remission. So, the concept that obviously made sense was to try to figure out whether if we combine both of those up front, that we might increase the likelihood that these patients stay in remission. And there's a number of ways that you can potentially do that. The way that we originally chose to do this was to make a single gene with a single set of signaling domains intracellularly, but then two binding domains in the same construct. So now this CAR can see either CD19 or CD22. And so when we infuse a product that has this receptor on the surface, if that leukemia tries to drop 19, there's another specificity that's in that receptor. That's at least the concept. And so we went through the preclinical development work for this. We published this in a series of papers. And this has actually now been taken into clinical trials at Stanford. And so far in the pediatric and adult trials, the remission induction rate seems comparable to both the 19 and the 22 CAR. It's a little early right now to know whether our hypothesis is correct and that we're actually going to see increased durability of remission. We've come up with other ways of potentially doing this with more complicated constructs. These are so-called bicistronics, in which we have two full-length chimeric antigen receptors in the same viral vector separated by these skip sites. And so we can then introduce both of these genes into the T cell. Now, instead of having one receptor, two binding domains, we have two full receptors, each of them with different specificity. And there's some very interesting signaling and immunology that goes along with this. So the other thing that I will tell you that we've learned in this trial, and it was predicted actually in one of our initial mouse models shown on the left, is that simple loss of antigen is not the only way that tumor cells evade these lineage-targeted immunotherapy. This is truly a lineage-targeted immunotherapy because we're going after a B-cell receptor. And you don't have to worry about the details, but that's called, on the left there, is essentially a gene expression profile of a mouse leukemia cell without CAR treatment on the far left there, and just look at the pattern of colors. And then when we give low-dose CAR treatment, there's recurrence, and it looks very similar to what went in, right? The problem is if we leave this leukemia in the mouse under CD19 CAR pressure, this particular leukemia, when it emerges resistant to the CAR at the end, looks very different than what went in. And what I can tell you is when you look at those genes that are modified in those recurrent leukemia cells, now instead of being a lymphoid leukemia, it's now a myeloid leukemia. It changes to a completely different lineage. And we published this in 2016, and about the same time, we started seeing the same patterns in humans with leukemia, particularly with subsets of leukemia that we know to be more stem-like, more plastic, so more able to modify their phenotype when we target specific lineages. And this obviously presents an additional challenge. So I just want to now in the last minute just talk a bit about this other issue of CAR T-cell failure. And I just want to come back to this notion that certainly this therapy seems to be very effective, but at its base level, it is an artificial construct. We are essentially piecing together different components of what are normal receptors in human cells, and then using them to endow specificity and activity of a T cell. But the details here don't matter, but just to point out that if you just look at the structure of the CAR and the T cell receptor, just at base, they're very, very different. You have much less in the way of signaling domains intracellularly in the chimeric antigen receptor. There's no organized synapse with the target cell. So we certainly are not recapitulating the biology of a T cell receptor. And we've certainly started to look at the signaling downstream of a chimeric antigen receptor and then compare that to the signaling downstream of a T cell receptor. And I can tell you that it is quite different. There are certainly similarities, but there are a number of differences that we think have an impact on how these cells behave in vivo. And if you just go back to a simple mouse model and ask a question of if you drop the dose of immune effector cells that you deliver to a tumor-bearing mouse and look at how efficient those tumors are cleared via the endogenous T cell receptor or via the chimeric antigen receptor, ultimately, in all of the experiments we've done, the T cell receptor is still more efficient. So I think there's a lot of opportunities to potentially modify these constructs to improve upon their biology. And so coming back to my first question, what have we learned? We've certainly learned, I think, that antigens comparable to CD19 are going to be difficult to find in other tumors. I don't think we're going to have that luxury, so to speak, of having an antigen that is dispensable. We've certainly learned that although there is comparable efficacy across all of those early trials with CD19, the details are certainly matter, and that's going to be more important as we move forward. We certainly know that tumor antigen modulation as well as more complex patterns of cancer resistance are going to emerge, and I actually think some of these are going to teach us some aspects of cancer biology that are going to be surprising. Current CAR formats certainly do not recapitulate T cell biology. But as synthetic receptors, the ability to modify is really almost endless. And so there's a lot of work being done in essentially taking these synthetic constructs and modifying them to try to endow specific properties. And I didn't talk about this, but I certainly don't want to be, end my talk without making a comment that certainly with the FDA approval of these cellular products, we certainly, no big surprise, are learning that the cost of and access to these sorts of complex therapies is definitely going to be a challenge, both in this country and certainly worldwide. So with that, I'm going to stop, and I won't, as is always the case, I do none of the work. These are the people who really do all the work, and these are the folks in my lab at the NCI, many of whom moved with me to my new home in Colorado. So thank you for your attention. I look forward to questions. Can you grab my water bottle? Just grab my water bottle. Yes, sir. Thanks.
Hi, Thank you very much for a great talk. Forgive me for a moment, but I'm going to try to bring together a little bit about what you said and what was said earlier this morning by Dr. Hahn. And I'm new to this, so bear with me for a moment. But what's really interesting to me is that the talk we had this morning was really about the immune system as a dynamic learning system within a context, and the context may be constantly changing. I think you said that in the conditions that you're looking at pediatric conditions that seem to emerge in this young population and emerge differently than in the older population and may be more virulent or have a bigger impact on that younger population. And so the question I would have there, I mean, there are all sorts of issues. For example, there may be differentiation within that population, et cetera, et cetera. But is it partly a function of the immune system not actually having learned sufficiently? And then, of course, going back to really what was, in some senses, the underlying theme in the first discussion, is there a way to train, and this is probably beyond it, to begin to train or expose the immune system in such a way that it does learn? And of course, the learning can have to do with lots of things, including how kids play and interact in the environment and what they're exposed to as they grow up.
Yeah, I'll make a comment as sort of a pediatrician, not an oncologist, but as a pediatrician in general, and as a parent who has kids who grew up in a time when the American Academy, as an example, the American Academy of Pediatrics, based on the high incidence of peanut allergies, made a recommendation to sort of increasingly to increase the age at which recommended exposure to peanuts should occur. And it was interesting. They, it was first it was 6 to 9 months, and then it was a year, and then they moved it out beyond a year, and it had no impact on the frequency of peanut allergies, and if anything, it actually went higher, I think, in some of the studies. And the current recommendation now has gone back to an earlier age of exposure, because I think one of the concepts, and this is certainly controversial, but sort of the hygiene hypothesis, so to speak, is that there's, the first year of life, infants' immune systems are sort of set to essentially try to figure out which things they're going to see for the rest of their life and to sort of turn those off in a way, because those are now antigens that they may be exposed to continually. And so that, I do think that there is a shaping of the immune system that takes place early in life, and it is probably not an accident that a lot of the malignancies that happen in kids happen in immune cells, B lymphocytes, T lymphocytes, because those are the cells that are being educated and rearranging at that time. So yeah, I think it's an important question, and there's certainly a lot we don't know.
Right in the back. Alan.
I think I'm on now. Could you speak a little bit about, I'm up here.
Up here. Sorry.
The use of these immunotherapies, particularly CAR T, in solid tumors, and what the issues may be and how you see us overcoming those?
Yeah, I think certainly there have been certainly lots of preclinical work, and more recently some clinical experiences to suggest that we may figure that out. There was some data this year at the cancer meetings about a CAR construct that targets mesothelin, which is expressed on mesothelioma, and really pretty dramatic responses. I think the challenge in solid tumors, there's lots, but I'll give you kind of two that I think are prominent. One is lack of good antigens. I don't think we're going to have 19s from any of the solid tumors. But the other thing kind of relates to the experience with immune checkpoint inhibitors, right? So most of those responses in immune checkpoint inhibitors are in solid tumors. And we know that solid tumors, as part of having kind of an organized microenvironment, are able to exclude immune cells or to shut immune cells down when they arrive at the solid tumor. So I have a feeling that in the case of solid tumors, it's going to be a matter of not just identifying a good antigen and making a good chimeric antigen receptor perhaps, but also focusing on these other suppressive mechanisms, combining with checkpoint inhibitors, et cetera, to try to address multiple challenges in the immune system.
I have a question for you based on the fact that bacteria, when they're dying, increase their mutation. One of the things they do is increase their mutation. Have you measured when these cells become refractory, or after 1 or 2 rounds of this pressure for dying, whether the mutation rate in these cells is the same low rate that you had in the beginning?
Yeah, so it's interesting. I mean, certainly, one of the thoughts has been that so cytotoxic chemotherapy, they're poisons basically, and they certainly, many of them drive, we think, can drive mutations in tumors. So one of the thoughts was perhaps in pediatrics that patients that had received chemotherapy that maybe a higher mutation rate might make them more likely to respond. It hasn't been the case so far, but certainly there's people that are interested in that, for sure. In the case of the evolution, if you will, the cancer evolution in the context of targeted immunotherapy, we've certainly done, pre and post genomic, epigenomic analysis of these cells. And even in the patients where there's a simple loss of CD19, it looks like that's an isolated event in the recurrent tumor cell. When you look deeper, the complexity of that evolution is certainly a lot more than you appreciate just by looking at the protein on the surface. So I think if you look at when we look at the patterns of epigenetic and genetic changes in those tumors, they're different than what you see in the context of cytotoxic chemotherapy. So I think, cancer cells essentially, modify themselves based on the specific pressure that they're seeing, and that certainly seems to be the case here.
But do you think it might go up enough to be sensitive to PD-1?
It's a good question. We certainly there are some trials now where people are giving, PD-1 inhibitors to patients who progress on CAR T cells, and really, the goal is to really try to look at that exhaustion, if you will, of the T cell, not so much based on the mutations of the tumor. But it's an interesting question. I actually think in the solid tumor world, we know that there are neoantigens because of these mutations in the solid tumors, and so one of the general thoughts is if you can get a good chimeric antigen receptor and you can drive inflammation in a tumor that's, for example, cold, right? That just having a single response, that will then result in what's called epitope spreading, and then you'll increase the diversity of the response based on the endogenous T cell receptors. And there's a lot of hope that might reduce that incidence of single antigen loss. So there's a lot happening that I think we're starting to figure out.
Okay, we have just time for 2 more.
Okay, one quick question. You may already have answered some of it already. I'm kind of curious, did anybody combine the checkpoint inhibitors at the same time with the CAR T cells simultaneously?
Yeah, so definitely is happening. I think if you go to clinicaltrials.gov, there's about 200 trials where people are giving checkpoint inhibitors plus CAR T cells because it was a logical thing to do. And I think one of the things we're facing here a little bit is when you look at the way that those trials were sort of designed and initiated, it wasn't really based on any kind of, predictive algorithm. I mean, it was just, we got one therapy, we have another therapy, let's just throw them together and try them out. And I think, I understand that that's typically how oncology, combinations develop. But, now that we have the portfolio of options are increasing, it's really impossible to do all of the potential combinations in the context of clinical trials. So that's where I think going back from those monotherapy trials and trying to learn from that experience and trying to generate this, what the next trial looks like, based on something that is, might be derived from that initial experience. I might predict what the best combinations are going to be. So yeah, people are doing it, but I think we're finding out that we're running out of, not running out of patients, but I think we're running out of certainly resources to conduct all the possible trials.
Could I just ask a little extension of that?
Sure.
So I want to make it easy for you by actually figuring out what I want to say. So those of us who've read a lot about both approaches, CAR T versus checkpoint, are kind of drawn to the idea that the checkpoint inhibitor program is one in which you need know nothing. You just say it'll happen. Whatever the antigens are, fine. And usually it will be the equivalent of your CD19, CD22 story. That is, it'll be multiples probably.
Yep.
And so you'll have less resistance,
Yep.
maybe, than you're seeing. And maybe you'll find out that the double 1922 will lead to longer or maybe no recurrence or something. So if you, instead of thinking about the impossibility of all the combinations, if you had to guess which path you were going to go down for all cancer, checkpoint and making tumors ready for whatever that means, irradiation or something, I don't know what it is. If you had to bet which, 50 years from now is likely to be the way we treat tumors, do you think CAR T has a chance to beat checkpoint inhibitors? That's my question.
Yeah. No, I mean, it's actually a great question. And I mean, I would sort of look at it from the perspective of, just the complexity of delivering the therapy and the cost, right? I mean, I'll say, the FDA approved, chimeric antigen receptor products right now, or, the bill is about $400,000. So they're not cheap, right? I mean, the checkpoint inhibitors are certainly expensive, but not at that level. And then there's the complexity factor. So, I mean, obviously we all, I think, would desire, an off-the-shelf, not a patient-specific treatment. The harder question for me is whether, for some cancers, whether we're always going to have to. I mean, I sort of look at the field of immunotherapy, in a very simple way, is really divided into two categories. There's immune enhancers, checkpoint inhibitors, you think of all those, and then immune initiators. And the question is, are there always going to be tumors where you have to have an immune initiator? I'm not saying I know what that's going to look like. It probably isn't going to look like the CAR that we have today, but I do think that for some tumors, we're going to have to do something to initiate that immune response for the reasons that I emphasize, that there just aren't antigens to be seen. Yeah.
We have to move on. Terry, thank you for a fabulous talk.